This is the beginning of Unit 2, Part 2. Proteins are extremely important and a varied, large group of molecules found in animals, in the cells, in the tissues, in humans, and it's extremely important to understand more about their structural organization. I remind you that at every level of an organism, at the molecular, the cellular, the tissue, the organ, and the whole organism, it's structure that subserves function. There's a particular things can do a certain function because of their structure. This is very true at the molecular level. And when we look at proteins, there are increasing levels of complexity that you can look at or think about the what goes into making the overall structure of proteins. At the simplest level, just the sequence of amino acids that are strung together in order to form a protein, that's the primary structure of a protein molecule. It's actually coded for in the DNA in a gene. Genes code for different proteins. Different genes each code for a different protein, and the code involves a code for which amino acid comes after, sequentially in the chain. That primary chain sequence of amino acids, in fact, is considered to be the primary structure. And then there's secondary structure. First we'll talk about primary. And then we'll talk about tertiary and quaternary. I'm going to move ahead. We'll talk more about this in the following slides. In terms of the primary structure, what you have is the sequence. And I should point out to you that this is a single strand is represented here in this diagram. It's cut off here, so it could extend much further down, but it's cut off here just to keep the diagram simple. What you see here are parts of three amino acids. You know it's an amino acid there because you can see the carbon in the R side chain, carbon in the R side chain, a carbon in the R side chain. So each of those all of this is one amino acid, and then this one is another amino acid, down to here, and then here's another amino acid. So they're held together by peptide linkages. The fact is that this is a representation of simple representation, and it's a classic biochemical model of a molecule. It's called a ball and stick model. And in this kind of model, different types of atoms, are represented by the convention is that a particular color represents a particular atom. Carbon, by convention, is black. So all of these are black. Oxygen's red, hydrogen is white. In this case they made it a light blue because otherwise it wouldn't show up against the white background. The R stands for the R side chain. Now a second level of characterization of the structure of proteins is secondary structure. At the secondary structural level, what you have in different parts of proteins quite often are repetitive, very stable, repetitive patterns of very stable regions. The regions are stabilized by excuse me. Each of these regions is stabilized by many hydrogen bonds within the molecule, and the hydrogen bonds in this case are shown by these dotted lines. So these hydrogen bonds that are found all over here in this example of secondary structure are all forming between an oxygen and a hydrogen. Remember, oxygen has a negative dipole often, and hydrogen has a positive dipole, so they are attracted and can form hydrogen bonding. Same goes for up here, hydrogen bonding all throughout this section of this molecule. Secondary structure refers to a particular pattern of stable formation within a molecule. In this case, what we have is a alpha helix, so it's curling around as a helix, or a helical, that's a better representation, a helical structure, stabilized by many hydrogen bonds within it. The other kind of uniform, stable structure that's often found in molecules is called a beta-pleated sheet. Sometimes it's called a beta conformation. And in this case, what you have, and it's shown in this diagram up here, and in this case, what you have here, in this case, what you have, it's not clear from the way the diagram is drawn, but what you have is, in fact, a single polymer strand of amino acids that loop back and forth on each other. So you have this single strand that's looping back and forth, and as it loops back and forth, it stabilizes into this secondary kind of structure called a beta-pleated sheet or a beta conformation. What's not shown here is the ends. In fact, this region here of the molecule, these areas up here of the molecule, are not shown in the diagram at the top. And so all you see are these straight line portions of the molecule, like this and this, running in parallel and interacting with each other by hydrogen bonding. And they form this kind of a pleated sheet going up and down, okay, as the parallel strands run beside each other. Lots of hydrogen bonding, a very stable portion of a molecule. Here's another representation. Here you see the primary structure, simply the sequence of amino acids one after another. And here you see an alpha helix, this kind of secondary coiling helical structure. Lots of hydrogen bonding, very stable. And here, the beta-pleated sheet. Remember that this, in fact, has a loop at some point, and it could be quite far away, and then, you know, often several parallel rows of it. Okay. The higher level of structural organization within proteins is at a tertiary level, so it's tertiary structural level. And in this case, what they're showing, and this is a non-classical artist's view of a molecule, but what they're showing is that you have a polymer structure, and the polymer strand goes like this. This is the primary sequence of it coming down to here. And within it, there are areas. Only one is shown, but there are areas of alpha helical structure. Here is an alpha helix in this area of the molecule here. So it does have some secondary structure. And the way the entire molecule folds in on itself and interacts with different areas of itself to form this kind of a stable formation, overall stable formation, is called the tertiary structure. Quaternary structure refers to molecules that are made up of more than one polymer strand. Now, it is possible to have, for example, two strands, you know, that are held together by some sort of covalent bond, in which case each is called a chain. The quaternary structure refers to the overall form and interaction shown here in this example of hemoglobin, of how it interacts, all the four parts of it interact. If the strands are held together by a covalent bond that I showed down here in yellow, then these are called chains. If it's by non-covalent bonding, then they're called subunits. That's a subtlety, but that's the definition. This example of hemoglobin, there are 4 polypeptide strands held together by non-covalent bonding, so there are 4 subunits to the polypeptide, to the overall protein. Each hemoglobin protein has at the center of its each of the polypeptide subunits a heme group, which holds, and you can see just a bit of one there and a little bit of another one. So each strand has a heme group in it with an iron that can bind to oxygen. Here you see a different artist drawing of tertiary structure. This molecule does have quite a bit of secondary structure. There's an alpha helical area here, an alpha helix here, and you see part of an alpha helix there, and then you see this beta pleated sheet area, beta conformation area, and then the overall folding back and forth of the molecule forms the actual tertiary structure. In this case, again, using the example of hemoglobin, they show the 4 subunits. You don't see in this case the heme groups, but they're there. And how these interact with each other and form the stable overall shape is called the quaternary structure. These you can ignore up here. They refer to another part of another diagram. Again, a different diagram. This is more of a conventional, there are conventional ways of drawing molecules and then artists' renditions. The ball and stick figure is a conventional way. This diagram here is a conventional way of showing the atoms that make up the molecule and their sphere, the van der Waals forces region. It's again hemoglobin. Hemoglobin has 2 identical subunits and beta subunits and 2 identical alpha subunits. And those 2 alphas are shown here, and the 2 betas are shown in green down here. You can see 2 of the heme groups within there. On the right-hand side, you see an artist's rendition of a hemoglobin molecule with the 4 heme groups inside, held within each of the subunits. So this shows, again, quaternary structure of hemoglobin. Okay, proteins overall can also be divided into what are called fibrous proteins. And it's important to understand that fibrous proteins are insoluble, so they're not in solution typically. These are insoluble proteins, and globular proteins, which are soluble. And globular proteins typically found, these are soluble, and they're typically found in the blood plasma or in the cytoplasm of cells. Okay. Fibrous proteins, some examples of fibrous globular. Some examples of fibrous proteins would be things like keratin, which is the protein that makes up fingernails and hair, or collagen, which is found in connective tissue like cartilage and tendons. There's certain fibrous proteins that have stretchy, elastic type of function. Some are contractile fibers that are found in muscle tissue. These are fibrous, insoluble. I should point out that the thing that they share in common is that they have a very long strand-like structure that has a lot of repeating secondary structure within it. Globular proteins, on the other hand, are quite compact. They tend to collapse around into a globular or round type of structure with many interactions. typically spherical, globe-like, a lot of tertiary and secondary quaternary structure, and some examples of that would be antibody molecules, the protein hormones. Not all hormones are steroid, some are protein. And an excellent example of globular proteins are enzymes, which very typically have this stable globular form. It's important to understand that overall, under controlled physiological conditions within the physiological range, that protein molecules will take on their most stable form. They do have a certain sequence of amino acids within them forming the polymer strand, but they will fold into stable areas of secondary structure like alpha helices or beta conformations, and fold further into tertiary structure. And if there's more than one subunit or more than one chain, they can form the quaternary structure. It will be quite stable. Of all the unlimited number of shapes that a long strand, polymer strand of amino acids could take, they have a very, very small number, and sometimes only one particular stable shape. This is the lowest energy level, typically the most stable form, the form that occurs in physiological conditions. And the shape gives the function. If conditions change from normal physiology levels, let's say temperature rises too high or too low, or pH changes too high or too low, then the molecule will change in its shape. The reason for these particular shapes under particular conditions is because each of the amino acids, I remind you, have a different side chain that has particular physical chemical properties. Some are positively charged, some are negatively charged, some are not charged. There are all kinds of, some are hydrophobic, some are hydrophilic. And so under certain conditions, they will have a small number of stable forms. And this refers to this kind of unfolding if conditions become non-physiological, not abnormal, too high or too low pH or temperature. The molecule can unfold, and that unfolding of the molecule is called denaturation. And with denaturation, what you get is loss of function. However, as long as you don't go too far out of normal range, as soon as the normal range is brought back, as soon as conditions are brought back to within the normal range, the molecule will fold back into its stable active form. So it's a reversible unfolding of the protein denaturation. In this case, it's showing an example of a protein shown here by this green spaghetti-like strand. I have no idea what these green globes or spheres, I suppose, are amino acids that are, or molecules that are forming hydrogen bonding to stabilize the form into this pattern. And it's an enzyme. Every enzyme has an active site. The active site is where it can interact with the, remember that enzymes are catalysts. They act as catalysts in biochemical reactions, and they speed up the reaction tremendously, the speed of the reaction tremendously. So every enzyme will have an active site where it interacts with the molecules that are going to take part in the biochemical reaction that are going to be converted into a product. So what we have are substrate molecules that will be acted on by enzyme catalysts to form a product. Conditions vary from normal, then there's unfolding or denaturation of the protein, the enzyme, and you lose enzymatic catalytic function. Here's another example of denaturation of a protein so that it loses here. There's lots of secondary structure, alpha helix. Here's an area of beta pleated sheet. So it loses that with denaturation, but it can regain it. It's a reversible process. It can regain it when conditions are brought back to normal. Again, here, protein denaturation. Sometimes that can be irreversible when you get to extreme changes in pH or temperature. Now, some basic characteristics of enzymes include the fact that they're typically globular, quite large in terms of the number of amino acids that they contain, but folded in on themselves are globular. They act as biological catalysts. They often, not always, need a cofactor to be active. They are very, very specific. They act only on their specific substrates because the substrate molecule they act on can fit into the active site. They are frequently named after the type of reaction that they act in, that they are catalysts in. Name often will end in ASE, and in general, the key thing is that they speed up reactions by lowering the activation energy required for the amount of energy required for the reaction to occur. And here's a diagram showing this kind of thing, that if you don't have an enzyme present, then the molecule has to gain quite a large amount of activation energy before it can go towards forming the final stable lower energy product. But if you have a catalyst present, that the amount of activation energy required is much lower. I can tell you that the real better presentation of how it works, in fact, is that the enzyme will bind the substrate and the product will be formed, and it'll require a lower amount of energy input to form the product, because what the enzyme actually does is it organizes the substrate molecules into a particular optimal orientation so that they can interact with each other and form the final product. The intermediate enzyme substrate complex has a particular structure, the two together here and here, but the final product gets released and the enzyme is free and able to go and catalyze exactly as it started, and has the ability to catalyze more reactions on more substrate molecules. Okay, let's move on to the fourth major class of molecules that are found in living systems, nucleic acids. Nucleic acids, you probably are aware, include DNA and RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. These are the information molecules that hold all of the information required to make an organism. However, I should point out that nucleic acids are not only involved in storage of information. There are many, many other uses for them, and I'll touch on one later. Nucleic acids are made of carbon, oxygen, hydrogen, nitrogen, and phosphorus. I've left out nitrogen here, but you could add it. Let's see, nitrogen. Right there. Okay. The basic structural building block or monomer of in nucleic acid polymers is a nucleotide, and the nucleotide is made up of a nitrogen-containing base, a pentose sugar, and a phosphate group. The nitrogen-containing bases are slightly different in DNA from RNA. The pentose carbohydrate, I don't like the word sugar, and we're not making tea or coffee, so it's not sitting in a bowl on your table. It's a carbohydrate. The pentose carbohydrates are in DNA deoxyribose, and in RNA ribose. So, as I said, a nucleotide, the building block for the polymer that will become a chain, a long chain of linked together nucleotides that form the RNA or DNA. There are 5 different nitrogen-containing bases. In DNA, it is A, G, T, and C. So A, T, G, and C. And those letters represent these nitrogen-containing bases. And in RNA, instead of T for thymine, we have a U or uracil. So RNA is made of AUGC. So ATGC and AUGC. DNA is a double-stranded helical molecule present in the nucleus of every cell. It is every eukaryotic cell. I won't explain that. It's present in the eukaryotic cell. It contains the genetic information that ensures continuity of life. As a cell divides and replicates, the DNA first is copied and then the cell can divide. And each daughter cell that comes from that original parent cell will have the exact same DNA. The double strands, the two strands that make up the helix, are intertwined with each other into a very stable kind of molecule. They are not covalently linked to each other, but they are bound to each other by hydrogen bonds. And if you recall, hydrogen bonds are weak, but when you have many thousands of hydrogen bonds between the two strands, forms an extremely stable type of structure. As I told you before, the DNA contains genes, and genes hold the information for the amino acid sequence within each of the different proteins that go up to that form a living organism. The carbohydrate portion of nucleotides, as I said, is ribose for RNA and deoxyribose for DNA. The difference between these two carbohydrates, 5-carbon molecules, is that on carbon 2 in the ring structure in ribose, you have a hydroxyl group here, whereas in deoxyribose, it does not have the oxygen, it is only a hydrogen molecule. What you see here is the kind of pairing that occurs between bases that form a part of each single strand. So this is one nucleotide, happens to be A, on one strand of DNA, on one strand molecule of DNA, and across on the other strand you will find always a T. They pair off always A with T and G with C, ATGC. So we're just looking here, in this case, at a single nucleotide shown here, phosphate, carbohydrate, and nitrogen-containing base, and on one long strand that goes up and down. And here the reciprocal or complementary base pair, which is a T always. If you have an A on one strand, you have a T on the other. If you have a T, you'll have an A on the other, etc. So there's the nucleotide, 3 parts, nitrogen-containing base, the carbohydrate, it's DNA, so it's a deoxyribose carbohydrate, and the phosphate, ATGC. Oh yes, I didn't mention it, but it's shown here also. There are, holding the two strands together are these hydrogen bonds, and they're shown by this these dotted lines. Please note that if there's 3 rows, that means 3 hydrogen bonds, and if there's 2 rows, it means 2 hydrogen bonds. Notice that between A and T, every A and T you have 2 hydrogen bonds, whereas between G's and C's you have 3 hydrogen bonds. So here are the nitrogen-containing bases, ATGC. They form, in a sense, if you look at it as a twisted ladder, they form the steps of the ladder, these nitrogen-containing bases, ATGC. The backbone strand, the polymer strand that winds back and forth of the helix, is made up of the deoxyribose sugar or carbohydrate and the phosphate. RNA is different from DNA. It's a single-stranded molecule. It is found both in the nucleus and in the cytoplasm of cells. And it, as I said before, it doesn't contain thymine, T, it contains uracil, and you get pairing of uracil with adenine, so it's AU rather than AT. There are three major varieties. There are actually more than three, but three major varieties of RNA. There's messenger, transfer, and ribosomal RNA, which we'll talk about later in the course. I mentioned to you earlier that although nucleic acids certainly are very important as information, genetic information containing molecules, they also serve other functions. There are other types of nucleic acids that perform other activities. A good example of that would be adenosine triphosphate, also known as ATP, which is a high energy containing molecule. The three phosphate groups, triphosphate, the three phosphate groups that are in the molecule, the bonds between the phosphates contain a great deal of energy, and cells are able to use this as a storage form for energy. They can make it, store it, and when they need it, draw on that energy. So it's a source of very usable, quickly used, quickly available energy, and it's due to the bond, the high energy bond between the phosphates. Here you see adenosine triphosphate molecule, ATP, the three phosphates in the phosphate groups, the ribose of the pentose carbohydrate or pentose sugar, pentose meaning 5 sides, the nitrogen containing base adenine. The nomenclature for these types of molecules is that if you cleave off one of the phosphates, then you have ADP, adenosine diphosphate. If you cleave off another, then you have only one phosphate. This one is left here, and you have adenosine monophosphate, AMP. And if you just look at the ribose and adenine, then it's called adenosine. Okay, so the whole thing is adenosine triphosphate. Here you see an example, some simple diagrams showing examples of how ATP is important. Certainly some molecules that are brought into the cell, this is the phospholipid membrane. You see a double layer of phospholipids. So it's a phospholipid bilayer. And there are membrane proteins that are involved in transport. So if a solute molecule needs to get transported in, it's the kind that requires energy, then ATP is used as a source of the energy. ATP can also be used as a source of energy for contraction of muscle tissue, or it can be used as a source of energy to break bonds in biochemical reactions. So transport, mechanical, chemical, it's a currency of energy for the cell. And that is the end of Unit 2, Part 2 on biochemistry.